Hello, everyone. Today we are going to be presenting the genetic basis of atrazine non-monotonicity in Drosophila melanogaster. Atrazine, a currently used herbicide, is typically applied on agricultural fields to combat broadleaf and grassy weeds. A variety of studies have shown that atrazine causes undesirable effects to numerous vertebrate species. For example, in male tadpoles of the African clawed frog species who were exposed to atrazine experienced demasculinization. In D. melanogaster, atrazine has caused negative, non-monotonic effects in studied traits such as egg laying, development time, and male reproduction. According to a study from 2015, male fruit flies had a reduced mating rate and reduced fertility when exposed to high levels of atrazine.
Non-monotonic effects or non-monotonicity is when traits develop a non-linear U-shaped curve. The purpose of this experiment is to determine the identity of the genes responsible for the non-monotonic characteristic. To accomplish this, a wide survey of polymorphisms in the fly genome were analyzed to determine if they contribute to atrazine non-monotonicity, as measured through egg laying. Although non-monotonic traits have been identified in fruit flies, little is known about the specific genes that cause this effect. The bar graph represents across all 30 fly lines the averages of proportions of eggs laid on food at each concentration of atrazine.
The results of egg-laying patterns for each fly line, along with each genetic identity, were put into a genome-wide association study, or GWAS program, which identified how variances in specific genes between fly lines associated with variances in egg-laying patterns. This identified genes indicated by the green dots on the Manhattan plot that are most likely to be associated with this variant, which were then investigated. As shown in the table, genes associated with the non-monotonic egg-laying distribution as related to atrazine susceptibility were found to be related to neurological, reproductive, and sensory development and function.
HBS1, which impacts sperm individualization and spermatocyte division, was correlated with our results. However, because only female flies were studied, future work may include male flies to further investigate the effects of this gene. PDFR, another correlated gene, has a known response to paraquat, an herbicide. A comparative study could be done between paraquat and atrazine to observe if there is a unique trait about the paraquat herbicide or if the gene affects responses to all herbicides, including atrazine. Future research could also be conducted around which genes have parallels in humans to better understand how these results affect us.